Hello, and welcome to this webinar, Cancer Immunotherapy, Where Will the Future Opportunities Be? My name is Rhys Armstrong, and I'm the editor for DDW and your host for today's webinar, which is being sponsored by BPS Bioscience and Acro Biosystems. The event will cover the biggest opportunities for the future of cancer immunotherapy, including the challenges facing the sector and the areas of research that are driving innovation. There will be a live Q&A at the end of this webinar, so make sure to stick around and post any questions you want answered to our speakers. During the webinar, you will learn about what is the next phase of cancer immunotherapy and what should researchers be focusing on to move to this next step. And where are the long-term opportunities and what challenges are there? In this webinar, you'll hear from immunologist Dr. Veronique Baron, CSO and CMO of Immunotherapy, two experts from DeciBio, Dr. Carl Scholhammer, Principal, and Dr. Joe DeCash, Senior Life Science Expert. Veronique Baron, Scientific Applications Manager of Marketing at BPS Bioscience, and Dr. Hengxiu Liu, Technical Sales Specialist at Acro Biosystems. Our first session will be presented by Dr. Veronique Baron, CSO and CMO of Immunotherapy. Veronique Baron founded Immunotherapy in 2001. He was appointed as Chief Medical Officer and Chief Scientific Officer following the acquisition of the company in December 2014 and joined Immunotherapy Limited's board in 2022. Before starting the company, he was Professor in Immunology at Paris University. While working at Institut Gustave Roussy, a cancer center in Paris, he discovered the LAG3 gene in 1990 and in subsequent research identified the functions and medical usefulness of this molecule. Veronique, over to you.
Hi. The topic of today is to talk about the future and in immuno-oncology. And first, I would like to look back at what happened in the last 10, 20 years, because there are maybe some lessons to be learned there. I've been working in the field for more than 30 years. In fact, my first paper on LAG3 was in 1990, a long time ago. And I've been working on T-cell immunology at the tumor site in patients. And I have a long time experience about cancer vaccines. So I remember 11 or 12 years ago, it was all about small cancer vaccine colloquium in the East Coast, and there were maybe 200 people there. Now, when you look at, for instance, SITC last November in Boston, there were more than 4,000 people, and many people from not only the academia, but big pharma, biotechs, and also all part of the industry. So it's all start with the registration of anti-CTLA-4, as everybody knows, in 2011, and then with anti-PD-1 antibody, namely nivolumab and pembrolizumab in 2014. At the time, the field named cancer immunotherapy switched to oncoimmunology, and then the year after to immuno-oncology. So, and now we call it IO field. And just after that, in 2015, we have seen the registration of the first IO-IO combination. In fact, it was two immune checkpoint inhibitors, two ICI, anti-CTLA-4 plus anti-PD-1. But the problem we have been facing in the past then is that between 2015 and 2022, last year, we haven't seen any new combination being registered. So last year it was anti-LAG3 plus anti-PD-1 register in first-line melanoma. And everybody recall all the failures associated with the field, the IDOs, and also anti-TIGIT more recently with quite a few problems there, that this combination didn't make it. And the question is why? I mean, why combination have not been a real success overall in the last 8 years? And maybe it's because we are insisting too much on a parallel approach. What is a parallel approach? Is, for instance, releasing two breaks on the T cells, like TIGIT and PD-1 or LAG3 and PD-1, meaning a combination of two ICI. And this leads us to think about not using a parallel therapeutic approach, but an orthogonal approach. Because when you think about this, you realize that a parallel approach could work in a hot tumor environment because the natural pre-existing T-cell immunity is quite high. There is a lot of interferon gamma, and therefore interferon gamma induce the expression of PD-L1 on tumor cells. And that's the definition of a hot tumor environment, at least in many carcinomas like non-small cell lung cancer or head and neck small cell carcinomas. So let's think differently. Let's think about out of the box and say maybe we should use an orthogonal approach and not a parallel approach. What does that mean, an orthogonal therapeutic approach? It's, for instance, combining a different class of product like APC activator. And APC activators have been around for a very long time. TLR agonists have been tested, CD40 agonist antibody have been tested, and more recently, eftilagimod, an MHC class II agonist, has been tested with some success in PD-1 antagonist, because basically it would make more sense in a cold tumor environment or in a tepid tumor environment, in patients with not so many T cells around, not so many activated T cells, not much interferon gamma. You may think about boosting the innate immunity part of it, the dendritic cells, while at the same time removing a brake on the T cells. And this could work not only in cold or tepid, meaning a low PD-L1 expression, but also possibly in refractory patients having received anti-PD-1 therapy and progressing because of primary or secondary resistance to anti-PD-1. And then what do you have to propose to these patients? And that's a real problem because when you pass the anti-PD-1 or anti-PD-1 plus chemo first-line therapy, you are stuck with very little. So again, to predict what could come out of that in the future, I would urge clinicians to think about basic science. What are dendritic cells? Why they are there? What are the T cells? In fact, dendritic cells, in my view, are the general of the immune system, because not only they chop the antigen, they give the antigenic peptide specificity, the T cell receptor specificity to the T cells. That is very well known, but they also give the direction, whether it should be an energy or T cell proliferation activation. And third, they also give the equipment to the T cells, especially to the CD8 killer T cells. They could give the license to kill to the CD8 T cells, the help. The help given to the CD8 T cells is not given by CD4, it's given by dendritic cells. And this is a key, really, if you want to have cytotoxic T cells and have an anti-tumor effect. So dendritic cells are mainly in the gut, the main secondary lymphoid organs. They are, of course, in the skin and all over the tissues. And one way to stimulate them is to trigger one of these port of entry on the surface of the dendritic cells, CD40, TLR, or MHC class II. So it's TLR receptors. They react to microbial compounds and may lead to some inflammation, while the two endogenous ligands expressed on activated T cells, namely CD40 ligand and LAG3, could activate dendritic cells directly through CD40 and MHC class II. So this is something one should think about for the next generation combination, whether you have to choose between a parallel or a orthogonal approach. And the orthogonal approach, if it works, of course, will have many benefits, especially in cold tumor environment patients. That's, I would say, is a key aspect of the current trend if we want to increase the potency of such IO-IO combination, or possibly IO-IO chemo triplet combination. So thank you very much.
Thank you very much, Dr. Treble. Up next, we have Joe DeCash and Carl Schulhammer from DeciBio. Joe DeCash is a senior life science expert at DeciBio. In his role, he addresses challenges for leading biotechnology in pharmaceutical companies, with a focus on next-generation therapeutics. He earned his PhD in immunology from Harvard University. Carl Schulhammer is a principal at DeciBio, where he leads the next-generation therapeutics practice vertical. He is co-founder of Sawano Bio and winner of a Lemelson MIT National Collegiate Inventors Prize and the National Collegiate Inventors Competition. Over to you.
Great. Well, thank you everyone for joining today. I'm Carl from DeciBio.
And my name is Joe DeCash from DeciBio as well.
And we'll be giving you a brief high-level overview of the field of cell therapies and their relation to immuno-oncology. This is really a burgeoning area with the approval of previous cell therapies and now the field moving to, as we call it, next generation cell therapies to try and do more exotic capabilities, ultimately to recognize and kill cancer for patients. This is an area we track very closely. We've run surveys with biopharma stakeholders in the past, for example, and this was just at the beginning of this year. And you see a really innovative area with a number of early stage companies, more mature companies, large pharma getting into this space. There's really positive sentiment around this modality class for a number of reasons we'll get into, but primarily the ability ultimately to treat a wide range of very difficult to treat cancers. So within biopharma stakeholders, you see, strong positive sentiments around likelihood to be able to raise capital to generate new programs in their pipeline, increase their headcounts, and most importantly, form partnerships with, larger pharma. And it all goes back ultimately to the science and people excited to see new data readouts of programs currently in development, greater personalization of these therapies. This is one of the incredible powers of these therapies and this modality in that it's ultimately personalized to the patient. And as a result, hopefully elicits even stronger and better responses and immune responses to recognize and fight that cancer. And also, as we'll touch on, novel technologies. So going beyond a cell therapy that simply recognizes one receptor on a particular cancer, but now those that are personalized to the individual, so they're recognizing neoantigens specific to that individual's cancer, as an example. Other modalities and functions such as secretion of other signals that can recruit additional immunostimulatory elements and different immune cells to also recognize and mount a response against that cancer, and even secretion of therapeutic molecules. So you could have a cell therapy that's, recognizing killing cancer while simultaneously also producing and secreting anti-cancer therapies as well, directly at the site of that cancer. I'll turn it to Joe to walk us through the pipeline.
Awesome. Yeah, and I want to re-emphasize what Carl was mentioning, the partnership which we're seeing in full steam this year, as well as another exciting area and topic in general that people are kind of watching is this new target discovery as well, where CAR T cells targeting novel antigens such as CD70, for example, which we've all heard really good, exciting news about earlier this year. So there are a lot of programs in development throughout the development stages, a lot of new cell therapy types, a lot of donor sources. You're going to see allogeneic, autologous. The move towards allogeneic, we believe, is slowing down a little bit. Autologous is still what is still promising, and at least for the foreseeable future for now. But in general, there are around 600 companies diving into the cell therapy space. The market is expected to hit around 15 billion by 2027. So it's a very sizable market and a lot of activity in the space currently. The majority of the products, at least to date, are still focusing on blood cancers, but you will see a lot of the activities actually moving towards solid tumors, A lot of hurdles in solid tumors, as you can expect, and not as straightforward as blood cancer, but you're seeing a lot of success in the field when people are investigating solid cancers specifically. Recent approvals for modified cell therapies ex vivo. So we're not only looking at, taking the cells, expanding them, putting them back in as raw cells, but it's more kind of we're modifying the cells. There is a number of programs looking at ex vivo modification through viral or even non-viral transfection techniques, which, there's a feel in the field that things are moving away a little bit from viral transfection towards a little more LNP-based or electroporation, mechanoporation, etc. So we're going to discuss that at the end of our webinar today. There are a lot of hurdles and QC/QA issues in cell therapy as similar to other markets as well, such as oligo or gene therapies, etc. But it's still on top of mind for stakeholders in the field, for CMCs. So there's a move towards tools that are minimizing some of those CMC issues, and we are also going to touch on it a little bit at the end. Again, big pharma is jumping in the space, as well as small biotech companies. You have Vertex, you have Bluebird, you have Astellas or Arsenal Bio, and on a daily basis, you hear news about, either a new program or new acquisition or something. There's not even one calm day in the field of cell therapy. We have exciting news coming out on a daily basis. Yeah, it's an exciting field. We're still early days. We don't have this 20% plus in phase 3 approximately. If I go to the next slide, this is the approximate distribution of cell therapies across the development stage. So there's a lot of focus on still early preclinical phase 1 products. I mean, as you expect, it's still a nascent, emerging, as Scott mentioned, a really burgeoning field. So you're not going to see a lot of approvals even. The products that are approved, the assets and drugs that are approved are actually pretty mind-blowing in terms of efficacy. But there's still this focus on expanding preclinical, maximizing the number of programs in phase 1, and kind of maximizing for the companies themselves, getting a product to the market. It's still a wide open field to kind of jump in the space and make a difference and get programs on. So I'll turn it to Carl to take us forward for the remaining of the slide, as well as the QC/QA part.
Yeah, thanks, Joe. So you're seeing with a couple of approved therapies now, a number of assets in late stage development. But this early pipeline, as we mentioned, really tackle remaining hurdles. And those would be around greater stimulation of the immune system, recruiting, a patient's own body to recognize and fight the cancer as well. Groups looking at new cell types beyond just T cells, which the field really started with as a cell therapy type. And then also going beyond, as Joe mentioned, blood cancers, going to solid tumors. And the ability to recognize a solid tumor and recruit the immune system to get an immune response there and ultimately not get exhausted. Because that's been one of the typical hurdles with trying to use cell therapies for solid tumors, has been that they get exhausted effectively. They lose their potency in that tumor environment because it's a low oxygen environment. And so these cell therapies just can't persist. So there's groups working on that to try and find, for example, better cell therapies that can withstand low oxygen environments, that don't get exhausted as quickly, and again, have a myriad of additional features and capabilities around secretion of novel molecules and things to stimulate the immune system, secrete anti-cancer molecules, while they're also engaging with receptors on the tumor type. And so one of the underlying hurdles of this entire field really goes to the manufacturability, the ability to take these cells potentially in an autologous format out of a patient, transform them, give them a new blueprint, and then put them back into that patient is quite a hurdle. And so the industry is focusing from a tool side on exactly that manufacturability. On the next slide, there's a whole ecosystem, and we'll call out that this is non-exhaustive, but a whole ecosystem of tools companies working on tools for both the individual steps of the manufacturing process, as well as the broader umbrella of manufacturability to automate the entire process end to end, so that you can put in, in an autologous format, put in the patient's own cells, get out the finished product to infuse back into that patient. So you see here groups that are working on one step on the left, and then all the way to sort of end-to-end features that would, spit out a finished cell therapy ready to go back into a patient on the right. And in addition to that end-to-end nature, what steps they address, also looking at the ability to decentralize this. So while large pharma historically has manufactured everything in large batches in a centralized, factory that then can be distributed out to patients, the thought here again around trying to reduce what's called the needle to needle time, the time from the cells being taken out of the patient to being put back in. And so decentralization is another important factor of that, in that you could potentially manufacture these therapies closer to the patients in clinical settings, in areas of excellence around major, oncology centers around the country or around the world. And so looking at how distributable is this equipment, are these processes, is another important element in the high level manufacturing of these therapies ultimately, to get them back into patients in a very timely manner ultimately.
Just to add, each cell type will lead to some tools that need to be tailored towards that cell type specifically. If you're working with NK cells or B cells, they are pretty much different than a T cell. So one tool for all cell types will not be the case, right? And again, to reemphasize, this is non-extensive. You have a lot of other companies such as Microfluid X, BPS Biosciences, a lot of Miltenyi Biotech products as well on the market. But at least it gives you an idea of how people are tackling some of this tool, and think about the tool space as like this enabler. Without it, things will take too long, the cost will be high, the vein to vein time will be really high as well. So the cell therapy will pretty much also be dependent and be enabled by those next generation tools.
Yeah, that's exactly right. If we look at the specific types of tests that are needed on the next slide, you see starting on the left, basic washing. This field, as it got going, really started to use tools that were legacy biopharma tools for developing other drugs, traditional biologics and things like this. And this is another area of need as the cell therapy field matures, is more purpose-built tools, like we showed on the last slide, for more basic things like QA/QC, your wash steps on the left, enrichment, taking cell populations from a patient and then further purifying those to find the best, most healthy cells to actually take down through the work stream and transform, making sure there's no bacteria in the culture, then activating these cells, looking at which sufficiently get transformed ultimately in the gene delivery step, which take up the material, and then expansion and formulation filling, ultimately moving from which cells have simply taken up the material to doing assays to look at which cells can actually now functionally recognize cancer and kill it. So that cell now will efficiently kill cancer before you put it back into the patient, as opposed to simply seeing, does the cell have the new genetic code that we wanted to deliver to it? And then in fill finish, a bit of work around activation and triaging a cell's ability to be exhausted or resist exhaustion. And so these are the critical types of tools that are starting to be developed and coming online to the drug development process to really help these therapies expedite their manufacturing, expedite their discovery, and get them more quickly into patients, and ultimately get higher quality cell therapies, into these patients. So here, looking again at that point around functional assays, while the field started, they were just triaging cells for phenotypic assays. Did they take up the genetic instruction we gave them? Are they now expressing, the chimeric antigen receptor, for example? To now moving to, rather than just simply, does the cell express that, to does the cell actually use it to recognize cancer and kill it? So looking at viability, and there's a number of groups that have sort of, typical assays for that. Cell proliferation, how well those cells grow and can expand. But now looking at target specificity in particular, and activation and cytotoxicity. So thinking about being able to triage and develop killing curves. So how well does that cell therapy in culture actually recognize and kill model cancer cells? How well does it resist exhaustion? How well can it withstand hypoxic or low oxygen environments? Those are the critical assays that are just starting to come online, really to help the field triage and better, select ultimately the cell populations to put back into people. There's companies like Excel Bio, developing tools to look for cells that can withstand hypoxic environments and therefore only select those cells to ultimately infuse and put back into patients. And it's those sorts of tools that will really help drive this revolution, drive the development of these therapies to ultimately make a positive impact, for these patients.
Yeah, and from a regulatory perspective as well, right, we don't see that data pretty much included on a mandatory basis in filings, but it's exciting to see how things will develop in the future. Those assays will be pretty important, not just from a competitive advantage, but also for products, but also from a efficacy perspective of the cells. Even, can they do what they are supposed to do? And if someone in one company is going to file that and the FDA sees that data and sees more efficacy, they're going to pretty much start requiring it as well. Another thing to point out here is the CDMOs and the CROs, especially early on, will play an important role in helping companies get that data, right? So we don't have tools that are, good to go, pretty much solid and implemented and installed in every company. So the biopharma and biotech companies will pretty much rely on CROs and CDMOs to be able to get things going at least before the companies start adopting those next generation tools, especially for the QC, QA, and inline QC, QA as well.
Yeah, that's right. So hopefully you found this high-level webinar informative, and thank you for attending.
Thank you.
Thank you, Joe and Carl, for that great presentation. Now we have Veronique Baron, Scientific Applications Manager at BPS Bioscience. Veronique Baron, who has a PhD and HDR, is a cellular and molecular biologist with extensive research experience in drug discovery and development, target validation, cell, signal and pathways, and cell biology. Dr. Baron has worked in a variety of research environments such as governmental and not-for-profit institutions, biotech, and pharmaceutical companies. She has published numerous patents and scientific manuscripts, has trained scientists at all stages of their careers, and for many years sat on grant review panels for the Department of Defense. Her technical experience includes cell culture methods, cell-based acids, 3D spheroids, flow cytometry, fluorescent microscopy, and more. Over to you, Veronique.
Good afternoon, and welcome to BPS Bioscience presentation focusing on chimeric antigen receptor or CAR cell development. CAR T-cell therapy has been a true breakthrough in oncology, and BPS Bioscience has been supporting scientists with their CAR development projects from very early on. So today, I will introduce our company and talk about custom CAR development services and tools for CAR cell development and functional validation of the CAR cells. BPS Bioscience is a scientist-founded, scientist-driven company established in 2005. Our mission is to advance new scientific discoveries leading to therapies by creating innovative solutions for research. We are supporting drug discovery in immunotherapy, epigenetics, SARS-CoV-2, and cell signaling. Our core technologies include the production of custom proteins and protein complexes, cell line development, CRISPR-Cas9-based genetic engineering, viral tool engineering. We provide screening and profiling services in which we test your drugs for you, and we provide pluripotent stem cell derivatives. Our custom core services are meant to help scientists from start to finish. This is a milestone measure process from the concept to CAR cell generation. First, the researcher provides the antibody sequence against the antigen of interest. We proceed with the engineering and validation of the scFv and test for specificity and affinity. From there, we produce the CAR lentivirus and perform an initial validation. If it works, T cells are prepared and transduced. We then functionally validate the CAR cells, and that includes CAR expression and cytotoxicity assays. Finally, the CAR cells are expanded and optimized. Now, aside from custom services, BPS Bioscience provides tools to support every step of the design and development process. And that goes from a fast, simple, quick PCR plus assembly kit for vector design, ready-to-use lentiviruses, cell lines and primary cells for CAR cell generation, recombinant proteins and antibodies to use in flow cytometry experiment to assess CAR expression. Cell activation can be facilitated using cytokine assay kits, CAR luciferase reporter cells, and one-step luciferase assay system. Finally, cell killing assays are facilitated by the availability of cancer cell lines, antigen overexpression cells, and luciferase reporter target cells. Now, to start with ready-to-use lentiviruses, our core and TCL antiviruses transduce pre-designed constructs against prominent immune targets, and that includes BCMA, CD19, CD20, CD22, and mesothelin. The antigen-specific TCR antiviruses transduce TCR constructs that recognize tumor-specific antigens such as MART1 and NY-ESO1. So these antiviruses are used to generate cells that express antigen-specific CAR or antigen-specific TCR cells. And these are for the design and optimization of bioassays used in functional validation steps. They can also be used as positive controls in any bioassay. Now, to generate the antigen-specific TCR cells, it is best to use TCR knockout cells. These can be generated using non-integrating or integrating TCR knockout CRISPR-Cas9 lentivirus. And of note, we also have TCR knockout cells already available. This flow cytometry data is an example in which human primary T cells have been transduced with a lentivirus expressing a CAR, bispecific anti-CD19, anti-CD22. So this shows the expression of the CAR. There is no CAR in the introduced cells, of course, and in the transduced cells, we see low expression and high expression of the CD19 and the CD22 CAR. Now, I just mentioned human primary T cells. Our portfolio of primary immune cells includes frozen human PBMCs that can be used with our cell isolation kits to prepare a cell population of interest. So on this graph, for example, I'm showing the enrichment in CD19 positive T cells. Then to accelerate our research, we make available negatively selected human CD8 plus T cells, as well as human CD4 plus T cells. And to accelerate your research even further, we have prepared CAR T cells that recognize CD20, 19, BCMA, and mesothelin. So CAR cell validation assays include co-culture assays in which the CAR cells are placed in the presence of the target cells. Activation of the CAR construct by the antigen induces the release of cytokines from the immune cells, and that can be measured using ELISA kits. So, for example, interferon gamma is released by anti-mesothelin CAR T cells when they are co-cultured with mesothelin CHO cells, and that's measured using the interferon ELISA kit for human interferon gamma. Now, these assays also use luciferase target cells, and that allows for very simple killing experiments in which the presence of the effector cells does not affect the measurement because only the target cells contain luciferase. Which brings me to luciferase expressing cells. So these are bioluminescent cells that can be used in vitro as targets in co-culture assays, but they can also use in vivo in animals as xenograft cancer models. These cells have been verified to be free of mycoplasma. They have low endotoxin. They are clonal, which means that they show uniform expression of luciferase, and that allows for very consistent readouts, and they're very affordable cells. We provide well-established, well-characterized human cell lines, including all these immune cells, and solid tumor cell lines such as colon cancer, ovarian cancer, and pancreatic cancer. The luciferase signal, of course, is directly proportional to the number of cells, that's shown here in vitro. And in animals, the luciferase expressing cells are detected by injecting a ready-to-use substrate solution. So as a conclusion, I will show an example of production and validation of primary CAR T cells. I have chosen here the anti-BCMA CAR T cells. These cells are produced by transduction using the anti-BCMA CAR lentivirus, the structure of which is shown here. CAR expression is measured by flow cytometry that can be performed using a fluorescent labeled protein, a biotinylated protein, or a tagged protein followed by an anti-tag antibody. In this flow cytometry, we have used a biotinylated BCMA followed by PE streptavidin. We show in red and transduced T cells which do not express the CAR of interest, and the transduced cells express high levels of CAR-C, whereas another population expresses low levels of the BCMA CAR. Functional validation is performed using cell killing assays. This can be done either with the BCMA luciferase CHO cell line or the Firefly luciferase RPMA cell line, which is a human cell line naturally expressing BCMA. The experiment shown here shows cell killing indicated by a decrease in luciferase activity, and this is proportional to the effector to target ratio. You will find resources on our website. The brochures contain product information and a complete list of products within a scientific category, whether it is cell systems, CAR T-cell therapy, immune therapy, and virus-based tools. Ebooks, tech notes, and posters contain details about specific topics. And this concludes my talks. Thank you for your attention.
Thank you, Veronique, for that informative presentation. Lastly, we have Dr. Hengshu Liu, Technical Sales Specialist at Acro Biosystems. Dr. Hengshu Liu has a background in cancer biology, cell and molecular biology, lung transplantation, and immunology. During his PhD, he participated in the Marie Curie ITN to study the function of cancer-associated fibroblast in tumor metastasis and invasion. Over to you, Dr. Liu.
My name is Hengshu Liu. I'm the Technical Sales Specialist from Acro Biosystems. It is my honor to join the DDW webinar. In today's presentation, I will discuss the challenge of cancer immunotherapy, how to overcome the obstacles, and the future opportunities. Cancer immunotherapy is a therapeutic approach for cancer treatment that uses patients' immune system to prevent, control, and eliminate cancer. It comes in several forms, including immune checkpoint inhibitor, T-cell transfer, monoclonal antibody, cancer vaccine, and immune system modulator. Cancer immunotherapy originated in the late 1890s when Dr. William Coley discovered that infecting cancer patients with certain bacteria sometimes resulted in tumor regression and even some complete remissions. In 1990, the FDA approved the first cancer immunotherapy, a bacteria-based tuberculosis vaccine called BCG, which was shown to be effective for patients with bladder cancers. During the past few decades, research has improved our knowledge of the mechanisms that regulate the immune system's response to cancer. However, challenges still exist, including the inability to predict treatment efficacy and patient response, the need for additional biomarkers, impaired efficacy due to tumor heterogeneity, the development of resistance to tumor treatment, autoimmune reaction, and high treatment cost. To overcome these challenges, researchers should focus on the following areas. Identify new targets that are specific to cancer cells and can be targeted by immunotherapy. Developing biomarkers that can predict and monitor patients' response to treatment. Improve combination therapy. Personalized treatment that can be tailored to each patient's unique tumor and immune system. develop next-generation CAR T cells that can target a wider range of tumor antigens and can overcome immune-suppressive microenvironments, promote cancer immunotherapy as an early administration to restore the anti-tumor function of patients' immune system. To better illustrate how to improve cancer immunotherapy, let's take CAR T as an example. So far, CAR T therapy has been only applied to hematologic cancer, which consists of 10% of the cancer diagnosis. There is an urgent need to expand CAR T therapy to solid tumor. Bioscience offers over 50 types of proteins, including hematologic cancer markers such as CD19, BCMA, solid tumor markers such as HER2, EGFR, PSMA. Furthermore, Bioscience has the platform to manufacture multi-pass transmembrane proteins, including CD20, GPRC5D, Claudin-6, Claudin-18, etc. The broader collection of CAR T targets will improve the cancer coverage of CAR T therapy. Expanding CAR T target is the first step for next generation of CAR T therapy. Clinical treatment with CAR T faces the challenge of antigen escape. In acute lymphoblastic leukemia, ALL, only 50% of pediatric and young adult patients treated with CAR T cells will be alive and disease-free 1 year after treatment. For patients with high disease burden at the time of treatment, the survival at 12 months dropped down to 31% due to CD19 downregulation. ALL patients who were treated with anti-CD22 CAR T cells after relapse with CD19 directed therapy achieved a 73% complete response rate, but 2/3 of these later relapsed with reduced CD22 expression. This challenge reflects the heterogeneity of cancer and how dynamic cancer cells are. Two strategies are proposed to overcome antigen escape, including co-administration of two CAR T cells specific for two different targets. The T cells can also be transduced with two CAR vectors encoding for two different targets, or transduced with one CAR construct with two single-chain fragment variable. These strategies can also overcome the challenge of tumor heterogeneity. So far, CAR T cells are engineered from patient-derived circulating T cells, so-called autologous CAR T. Although avoided of cell rejection, autologous CAR T limits collection efficiency, which is one of the reasons for its high cost. Next-generation CAR T therapy aims to expand the T cell source by establishing allogeneic CAR T. The T cell source will include circulating T cells from healthy donors. T cells differentiate from iPSC or stem cells isolated from umbilical cord blood. These allogenic T cells pose a risk of causing T cell graft rejection and graft versus host disease. A potential solution is the removal of TCR and beta microglobulin by improved gene editing technique. Among the allogenic T cell source, iPSCs have great potential in cell therapy for their multilineage differentiation ability. Besides of T cell differentiation, iPSCs are also used for nitric killer cells and macrophage differentiation, which are important sources for CAR-NK and CAR-macrophage that will further improve cell therapy options. iPSC culture requires critical microenvironment that can be formed by base membrane extract. Base membrane extract is collected from extracellular matrix-enriched mouse tumor cells. It is composed of laminin, type 4 collagen, nestin, glycoprotein, and different cytokines. By modifying the components, base membrane extracts provides different microenvironments for iPSC expansion and differentiation. For its fundamental function in iPSC culture, basement membrane extract will play an important role in next-generation CAR T therapy. Following the workflow of CAR T therapy, after identifying the target, clone the CAR, isolate the T cells for CAR engineering, the next important factor for CAR T therapy is cytokines, which is indispensable for CAR T cell expansion. During this step, engineered CAR Ts are expanded in the lab to millions of identical copies that are then infused back to patients for cancer therapy. Cytokine products are required at different stages during CAR T development. Acro Bioscience provides different grades of cytokine for specific needs. The RUO grade active max products are manufactured at ISO 9001/13485 certified facilities with research grade materials. Final products are sterilized by filtration. They are cost-effective for exploratory studies during research and development phase. The premium grade products are manufactured by the same production method of GMP grade. They are animal origin free and have comparable quality to GMP, and have similar pricing to RUO. These products can transit to GMP grade seamlessly and are suitable for preclinical phase. The GMP grade cytokine products are required as raw materials for GMP grade CAR T expansion. Acro Biosystems provides strong support on cell therapy development with our product. We have broad coverage of target proteins, including more than 50 CAR T targets. We provide cytokines, activation beads, and basement membrane extract for ex vivo CAR T activation, expansion, and modification. Moreover, our antibodies and kits are powerful tools for detection of CAR expression, residue cytokine, and nuclease. In conclusion, cancer immunotherapy is a powerful and promising clinical approach, but facing many challenges, including unpredictable efficacy, developing resistance, high cost, etc. Future research should focus on improving cancer immunotherapy by enhancing safety and efficacy, improving combination therapy, and identifying new biomarkers. Thank you very much for listening. I'm happy to answer your questions.
Thank you to everyone for those fantastic presentations. Before we continue to the Q&A part of this webinar, I'd like to remind everyone that this webinar will be available to access on demand on the DDW website, www.ddw-online.com. Could I invite all our speakers to turn on their microphones and webcams, please? Okay, thanks everyone. So we'll move on to the questions now. I think we'll start with this first one for Veronique. You've seen great strides made in the cancer immunotherapy field during your career. What do you think will be the most significant advancement in the field within the next 5 years?
Well, let's talk about the past. Indeed, I've seen many strides over the last 40 years. The most significant one for advanced cancer carcinomas was in 78, 79, where we have seen anthracycline, new class of chemotherapy, and with that we were able to treat and maybe cure testicular cancer, half of ovarian cancer, Hodgkin disease, and that was really a great progress. Now, in between, we have seen the taxane, we have seen other chemotherapy, but not changing really the field of advanced cancer. More or less, when you have stage 4 carcinoma, you are not doing well, and in terms of overall survival. And 10 years ago, we have seen the PD-1 antibodies, and for, of course, a revival of cancer immunotherapy, which is now called Immuno-Oncology. And but this is about 20-30% of carcinomas, and of course there's a long way to go. The good thing is that we learn along the years, and in fact, when you have a partial response at 6 months following PD-1 therapy, you are doing good. At 2 years, 3 years, you will be in remission, meaning that if you have a good CD8 T cell response that has been effective because you get partial response at 6 months. This is sustainable, even so you stop PD-1 therapy. So it's different from TKI or chemotherapy, where you see very quickly resistance, secondary resistance to the drug, and the tumor coming back. So it's really, we are learning along the years, but it's a very, very long way. I remember the 80s. In the 80s, we were doing at our institute in Paris bispecific antibody. One arm is CD3, the other one with a tumor antigen, trying to get killer T cells together with tumor cells together. We were growing TILs for melanoma. We are growing purified NK cells, mass expansion, and we are trying a lot of things. And in the last 30 years, I mean, unfortunately, cancer vaccines, meaning specific active immunotherapy or cellular therapy, passive immunotherapy, did not work so well. So it was kind of a miracle that in 2014, we have seen these great autoimmune disorders appearing in metastatic cancer patients, meaning that at least one immunostimulant is powerful enough to induce autoimmunity, which is expected in a way. And these patients as well had a huge regression of the tumor size. So that's the way clinicians learn how to use these immune checkpoint inhibitors. And indeed, if you look at non-small cell lung carcinoma, the biggest market, many patients do not receive chemo anymore, and that's something really striking.
Mm-hmm.
So
Great.
what will happen in the next 5 years? I think it's all about combination. I mean, combination have been there for very long. I mean, in terms of epistemology, if you look back, they were there in the 60s, because we learn as hematologists that adding a second chemotherapy on top of the first one in lymphoblastic leukemia in children, you double the response rate. So, of course, combination are there. And as always in advanced cancer, especially in first line, you have to use the standard of care and add something on top of it and see what it does. And I think active immunotherapy is the way to go. Of course, it would be very nice that to have cancer vaccine one day. We have been working with that, different approaches over 30 years. But now, maybe with the mRNA approach, where you can select 40 neoantigen in a tumor in lung cancer, and you use mRNA, maybe you could get strong enough CD8 T cell response specific for these neoantigens, and together with PD-1 see something. But it's a very long way where we don't know how to get a good tumor response in primates. I mean, cancer vaccine have been curing rodents for 30 years. It works beautifully, but not in primates, not in human. So we are very different. And maybe we will see that, and I hope I will see that, because I've been working on cancer vaccine for a very long time without any success before.
No, absolutely. Thank you, Veronique. We have a question for the whole panel now. Do you know where I can find a monoclonal antibody listing used in cancer and inflammatory diseases?
Sorry, what exactly is your? I don't get the point here. Sorry.
So I think we have an audience member asking where they can find a monoclonal antibody listing used in cancer and inflammatory diseases.
I would just go to Abcam. There's a number of antibody repertoire companies: Abcam, Bethel, they'll have a good repertoire and sort of menu of relevant antibodies that can be used in this work.
Great, thank you, Carl. I hope that helps. Carl, we'll move on to a question for you, actually. What are some of the bleeding edge trends you're seeing in cell therapies, just generally?
Yeah, I think what we're seeing is a move, beyond just chimeric antigen receptors and CARs to really these cell therapies that have additional functionality. So they have a CAR, they might secrete additional immunostimulatory cytokines. They might secrete actually directly, PD-1 antibodies or other, anti-cancer, basically therapeutics. So now you have a CAR that is also a drug factory, that can resist the suppressive effects of a tumor microenvironment, and really all with the goal of moving cell therapies towards solid tumors, beyond just blood cancers.
Amazing, thank you. Heng Shu, how does cancer immunotherapy differ from other therapeutic approaches?
Well, I think the main difference is that if this therapeutic uses the host immune system to cure the disease, I think that's the one major difference. For example, this CAR T, immune checkpoint inhibitor, etc. They all are aimed to enhance the immune system to destroy that. There are some other, let's say, treatments. For example, ADC, antibody drug conjugate, is not considered as cancer immune therapy, but it's also very powerful approach for cancer treatment. I think this definition just to basically define that what we talk about for like therapeutic approach, but it doesn't really set like the priority or which one is more favorable for the market.
Yeah. Great, thank you. Veronique, we have a question here for you. What are the advantages and risks of using lentivirus transduction compared to, say, non-viral based methods such as transfection?
Yes, good question. I think the main advantage for lentiviruses really is efficiency. If you're going to introduce genetic material with a lentivirus, you'll have a much higher percentage of cells that will be transduced compared to transfection. But it also, transfection tends to be very efficient in cells that proliferate pretty fast. And so for non-proliferating cells or for primary cells, for example, lentivirus is going to be much more efficient. The other aspect of it is if you're trying to do a stable cell line, like to generate a stable cell line, well, lentiviruses really have evolved to introduce their genetic material and have it integrated into the DNA. So they are much more efficient at doing this than transfection is. So to generate a stable cell line, lentivirus is much better. I think they also can introduce larger DNA cargo. I think the lower toxicity of transduction as opposed to transfection is another aspect to consider. It's much less toxic to the cell. So if your cells are a little fragile or if they're very sensitive to lipid-based transfection, you're better off trying with lentivirus.
Amazing, thanks. And Joe, just moving on to you, what do you think the potential impact of decentralization manufacturing could be on patients, payers, and clinicians?
Yeah, great question. So currently, when someone wants to manufacture cell therapy, especially on autologous side, you do need to ship your cell collection to a manufacturing facility. Could be geographically very not located in an optimal way because it has to be centralized. So the potential of decentralized manufacturing could decrease cost. For example, it could decrease time, so vein to vein time. Could decrease some of the supply chain hurdles that physicians and patients encounter as well. So on patients, you'll see something, a faster time to therapy, potentially lower cost, especially people who are uninsured or people who have low coverage of insurance. That is a little more impactful in the US than EU, for example, but that's still a consideration in terms of cost. terms of payers, I mean, that the decrease in cost will probably be an attractive value proposition for payers as well. And that's going to be a driver for covering more and more cell therapies on the market. And for clinicians, I mean, we also have to keep remembering that physicians are not, they don't have a patient and forget about the patient and wait for the therapy to come back to them. They also they get pretty anxious because those patients are sometimes in extreme conditions and they do sadly still die because they are waiting on cell therapy. So this decentralized manufacturing will allow faster cell therapy manufacturing, a little more local, accessible manufacturing, and get those therapies faster to patients at the end.
Great, thank you. And Carl, just another one for you. What are some of the key bottlenecks in the development and production of these therapies?
Yeah, it really goes to the engineering of the cells. One of the other panelists mentioned lentivirus is commonly used today for the transduction efficiency, but groups, because of potential toxicity risk and incorporation risk and things, are trying to move to non-viral methods. So what efficiency can you actually achieve of the number of cells that are engineered to then expand those cells that were effectively engineered, grow them up, and then doing all the QC/QA assays and things. And so there's a number of groups, as Joe mentioned, working on automated methods, or sort of all-in-one production methods, particularly for autologous-based therapies, where it would be, one box per patient treatment effectively. But that's the piece we're getting around and trying to expand up then the population of engineered cells to put back into patients.
Amazing, thank you, Carl. So we have a question for everyone here by somebody in the audience. CAR T relies in a large part on the antibody recognizing the tumoral antigen. Do you think the quality of those antibodies and their affinity for the target might have an impact on the overall efficacy of the therapy, or in this case, is simply binding enough for a good CAR T therapy?
Well, I'm not a specialist in CAR T cells, but I can comment maybe a little bit on this. My understanding of it is obviously the quality of the antibody is super important. If you're going to target a molecule that is present on a tumor cell, but it's not supposed to be present on other tissues, for example, you don't want your antibody to cross-react with any of these other tissues. That's one aspect of it. But in terms of affinity and avidity, there are new attempts now at making CAR T that are actually a little weaker in affinity, and they have been giving very good results. So better affinity is not always better in terms of efficacy. And there are quite a few studies now that are looking into that area where you want something that is highly specific, but affinity may not be the best criteria for a CAR T cell. Anyone else wants to add something to this?
No, that's absolutely fine. Thanks, Veronique. We do have another one here for the whole panel. If somebody could they give a short comparison between T-cell engager bispecific antibodies versus CAR T cell therapy?
Well, maybe I can just have a comment on this. CAR T comes from the TIL experience, from the TIL, what has been done before, different countries, Stephen Rosenberg in the US, for instance, in the 80s, 90s. And bispecific have been used also widely because it's both are very old technologies. And not much progress have been made. And in fact, they are kind of passive immunotherapies, and passive immunotherapies are in general more expensive and maybe not there forever. It's a sort of an interim approach where very useful because you learn a lot. But at the end of the day, it may be replaced by active immunotherapy. which is a more direct. And for instance, we learned that in the early 900 with tetanus toxoid, we were immunizing horses during the First World War, injecting the serum. And then in 1922, we used the tetanus toxoid with aluminum hydroxide as a vaccine and solved the problem with a specific active immunotherapy and not using the serum anymore. And so that's the way it should be. Unfortunately, many active immunotherapy approaches have been very, very difficult, including therapeutic vaccines in patients. And CAR T cells are not easy as well. I mean, it's working with CD19 more or less. And so it's hopefully in 30 years from now, there will be good active immunotherapy and all these problems will be solved. That's my hope. So it's much more than beyond this next 5 years.
Absolutely. Thank you, Veronique. And I think we've got time just for one last question. Dr. Henshaw, how can we make CAR T treatments more accessible?
So the current biggest problem with the CAR T therapy is that the cost is way too high, and there are several reasons for that. First is that the manufacturing scale is very small, and now the approach is that so you harvest the T cell from the patient, engineer into CAR T, then put it back. So that limits the manufacturing scale. So, and therefore, in my presentation, that the proposal for the next generation CAR T, we need to look for other source, like for example, to use people allogenic donor for the CAR T, or use stem cell differentiation to do that. Of course, we face challenges to achieve that, and I think these are still under R&D. But the major goal is that because CAR T is very powerful skill, and for cancer treatment, we cannot really rely on one single therapy. We need a combined therapy. We will want to have our handful of options in hand, and then reducing the price, it's something that we have to achieve. Yes.
Amazing, thank you. And I'd just like to invite all our other speakers to see if they have any thoughts, quick thoughts on that answer given by Henshaw.
Actually, yes. From the question of the BSCB, it gave me some idea that when I think the difference between the BSCB and CAR T, the CAR T, the purpose is that the T cell is already activated, engineered, ready to recognize and attack. But BSCB is about, if I understand correctly, is about to bring the T cells and tumor cell close together. But in this case, how to ensure that this T cell actually can recognize and attack the tumor cells through my immunology background, that T cell need to be activated. But that's something actually I need to do some homework to be able to answer the client's question about that. Yeah,
that's absolutely fine. Thank you. So with that, it brings us to the end of the webinar. I'd just like to thank everybody for tuning in, and once again, a big thank you to Frederic, Carl, Joe, Veronique, and Henshaw for once again taking part. Just to our audience, if you would like to listen to the webinar again, it will be available on demand at www.ddw-online.com. Thank you, everyone, for taking part.
Thank you.
Thank
It
you.
was a pleasure to be here. Bye-bye.
Bye-bye.
Thank you to the sponsors of today's webinar, BPS Bioscience and Acro Bioscience. You can now view Drug Discovery World content online on your mobile or tablet, or via the new DDW app, which can be downloaded via the App Store or Google Play. Thank you for listening.